Hi scholars, today we're going to discuss microscopes, magnification, image size, and actual size. And this video will help you understand how to do the calculations on the last page of the Microscope eLab. So, firstly, when you look at a microscope image, you are looking through the eyepiece, and you get 10x image there. And when you look through the high power objective, you get another 40x. So the total magnification, as you remember from the lab, is going to be 400x, 10x times 40x. So if I had a dot and I was looking under 400 magnification, it would look 400 times bigger. So if it was 1 millimeter diameter in real life, it would be 400 millimeters in the image life. So we have to come up with a calculation to help us understand the relationship between these numbers. So we have our actual size, we have our image size, and we have our magnification to get us between our actual and our image. So we think about this for a second and say, okay, what I'm doing is I'm taking my actual size, I'm multiplying by my magnification, and I'm getting image. And we can solve for m, our magnification, and we get image over actual. And if you look on page 12 of your book in the top left hand corner, you can see that this is the equation that you should have already written down in your notes that I've collected and will be giving back to you soon. So before we take a look at the questions on the last page of the Microscope eLab, I wanted to share with you a few sample IB questions to help you understand the kind of knowledge that you need to have. The diatom Didymosphemia geminata is a species of single-celled algae that lives in warm, shallow water. In the light microscope image below, the scale bar is 10 micrometers. You see this bar right here. What is the actual length of the cell? So what you're supposed to do is just take a ruler and realize that this bar will fit 7 times across the length of this cell. So we take this bar and multiply by 7, and we'd get 70 micrometers. Now you notice 70 micrometers is not one of my answers. I actually have to convert it. So there are 2 ways to do this, and I'll show you quickly the dimensional analysis way that I hope you learned last year. You need to know that there are 1,000 micrometers in 1 millimeter. That's just something you have to know. So remember that a milli is 10 to the negative 3 meters, and a micrometer is 10 to the negative 6. So there's a 1,000-fold difference between milli and micro. So you want to cancel the micrometers, and you want to get millimeters. And you see that there's a 1,000 next to the micrometers, so you put 1,000 at the bottom here. And a 1 is next to the millimeters, so you put a 1 at the top. And you want to solve for millimeters. This method is called dimensional analysis. You learned something like this when you're doing stoichiometry last year. And so you notice that the micrometers cancel to get the unit that you want in your answer. And when you divide by 1,000, you move the decimal point 3 times to the left. You don't need a calculator. So we move it 1, 2, 3. And you get 0.070 millimeters. And you see that the answer is B right here. So let's take a look at another question. A student observes and draws an amoeba using the high power lens of a microscope. The diameter of the drawing is 100 millimeters. The actual diameter of the amoeba is 100 micrometers. What's the magnification of the drawing? So remember that magnification is the image size divided by the actual size. So as I see, the diameter of the drawing is 100 millimeters. So that would be the image size. The actual diameter is 100 micrometers, is right here. Now notice, they're different units, so you can't possibly divide them if the units are different. So you need to convert one of them to the other one. So we could go through the calculation that we just went through. I'm just going to convert the top one to micrometers. Put millimeters on the bottom to cancel it, and micrometers on the top, so that's what we want. And we know then that there's 1,000 micrometers in 1 millimeter, and that means you have to move the decimal point 3 times to the right. So there's my 100, and I add 3 more decimal places. So going back to the problem at the top here, I have 10,000 micrometers divided by 100 micrometers, and you see that 2 sets of zeros cancel out, and my magnification is going to be 1,000, which is the answer right here. Now there's another way to do this calculation that I've shown you at the bottom, and that is kind of a little shortcut. If I want to go from 100 millimeters and I want to know how many micrometers it is, well, I have to know that when I'm going from millimeters to micrometers, my unit is getting smaller, so my number has to get bigger. Think about when you go from dollars to cents. When you go from dollars to cents, you know that the unit is getting smaller. But when you go from dollars to cents, you know you're going to have more cents. So you have to multiply by 100 because there's 100 cents in a dollar. Well, it's the same idea here. The unit is getting smaller, so the number has to get bigger by that number of times. So that's 1,000 times bigger. So I'm going to move the decimal place 3 times to the right. We'll try it again with the next problem. If a red blood cell has a diameter of 8 micrometers and a student shows it with a diameter of 40 millimeters in a drawing, what is the magnification of the drawing? So magnification is the image size over the actual size. The image size is 40 millimeters and the actual size is 8 micrometers. Now, just to prove to you that we can change the bottom one and get the same answer, we will take micrometers, 8 micrometers, and convert it into millimeters. So we notice that the unit is getting bigger, so my number has to get smaller by 1,000. So that would make it move the decimal point 3 times to the left. 0.008. So it would be 40 millimeters divided by 0.008 millimeters. The millimeters cancel, and you notice that when 8 goes into 40, it would be 5. So note that the answer has to have a 5 in it. And it can't be 5 because there's no way that 0.008 that's only 5 times into 40. So the answer is 5,000. So if we take a look at our last page to Microscope eLab, we have our equation that goes at the top. Magnification is the image size over the actual size. And if we scroll down here, we can see that this bar is supposed to be 10 micrometers. And I measured the size of this bar with one of your classmates. I measured the length of this. And we found out that the length of this in the image is 30 millimeters. And the length of this image using this bar is 33.3 micrometers. So the idea is that we have to make it the same unit. So I'm going to convert the top unit to micrometers. keep the bottom one the same. And so when you convert millimeters to micrometers, you know the unit is getting smaller, so the number has to get bigger by 1,000. So now we divide these two numbers and we would get our magnification. I'll let you do that part and then let you practice these other problems. So I hope this helps a little bit in helping you refresh your memory on unit conversions, but also to understand how to use this equation up here.